Good morning. The major consideration of the unit chromatography for the student is at the end of the unit the students can able to know what is the principles of chromatography. The next one, the instrumentations used in different chromatographic techniques and understand the applications of chromatography in various fields, and also they can able to perform basic chromatographic experiments and interpret the results which has been used in our day-to-day lives, and they can compare and contrast different chromatographic techniques. Also, the major key consideration of planning is the first one, they can have the simple concepts and gradually move to the advanced techniques. And the second one is they include the experiments and consideration to reinforce the theoretical concepts. And the third one is the real world applications. They are using the concepts of chromatography to everyday life and industrial uses. The fourth one is the interdisciplinary approaches, which is called as the link, the chromatography of biology and chemistry, environmental science, and the forensics, which has been the crime scene analysis used in the day-to-day life of the police and CBI functions. And the assessment varieties of using the formative and summative assessments to evaluate the understanding functions. And the engagement strategies, they can incorporate the interactive and the collaborative activities to maintain the students' interest. So the major introduction of the chromatography was they can use the technique to separate a compounds of the mixture based in the day-to-day functions. They can have a demonstration. They can use a simple paper chromatography to separate the inks and the leaf pigments. They can able to easily understand what is called as a chromatography, and the simple chromatography was the first is called as the paper chromatography. By using the simple filter paper and the solvent, they can separate the analysis of the ink and the different variations of colors by using the gems and also the pigments of the leaves. And the second one is using the column chromatography. In the column chromatography, we can use the silica gel and alumina and the mobile phase of the solvent. We can able to separate the different compounds as per the purification process. And as for this, we can able to use the activities for the students to separate the food dyes by using a minimum column functions. And the third one is of the ion exchange chromatography. By using the charge interactions, we can able to separate the ions which has been present in the solutions. And this has been used in the applications of water softening, because in major things we can able to have the hard water. By using the ion exchange process, we can able to separate the hard water into the soft water, and also the protein purification processes. These can be explored in the biochemistry functions. The removal of the calcium ions from the hard water by using this ion exchange process. And by using thin layer chromatography as a solvent, we can able to have the various polarity of the substances has been separated by using the thin layer chromatography. And all these things we can able to calculate the RF values. And by using the gas chromatographies, we can able to have the inert gas. By using the inert gas, we can able to separate a large number of gases separated, and this has been used for the forensic analysis and also in the environmental monitoring processes. By using this, the virtual labs, we can able to have the gas analysis of volatile compounds. In high pressure liquid chromatography, we are using this in major researches for the development of the drugs and for the food safety testings. By using all these things, we can able to make the students to know the major advantages of using all these things in the day-to-day lives. By using the engagement strategies, we are having group activities, the databases and the case studies, the chromatographic techniques, the themes and the quizzes. The students can be able to easily interpret with this in the day-to-day lives. These are the importance of the students to have this chromatographic for the units. Thank you.